Hello, my name is Leisa Jackson, and I am a scientist on the assay development team at Biodesix in Boulder, Colorado. The title of my talk is T-cell receptor sequencing in liquid biopsies from NSCLC donors. Just a brief outline. First, I'll start out with an overview of Biodesix. Then I'll go into some background on immune checkpoint inhibitors, as well as how T-cell receptor sequencing is used as a predictive biomarker for immunotherapy. And then I'll talk a little bit about some of our in-house T-cell receptor sequencing studies that we've done. First of all, looking at peripheral blood lymphocytes from NSCLC patients, and then I'll talk to you about a model antigen study that we did using cytomegalovirus antigen. So Biodesix has two core business units. On one side, we have our lung-focused diagnostic testing business, where we work with physicians to help solve unmet needs in lung disease. On the other side, our capabilities for biopharma partnering are disease agnostic and include diagnostic discovery, development, and clinical trial testing. All of our testing is performed within one of our two high complexity laboratories. Our CAP-accredited CLIA, New York State CLEP, and ISO 13485 certified lab in Boulder, Colorado, or our COLA-accredited New York State CLEP certified lab in DeSoto, Kansas. We provide a portfolio of 6 diagnostic tests for lung disease. We have 2 blood-based diagnostic tests that support lung nodule risk assessment as part of our Notify lung testing strategy. And we have 2 blood-based diagnostic tests that support treatment guidance for patients with lung cancer as part of the Biodesix lung reflex strategy. More recently, we launched 2 diagnostic tests as part of the Biodesix WorkSafe testing program to provide testing for the COVID-19 pandemic, a viral RNA, ddPCR test, and a serology test looking for total antibody. Our testing platforms support a multi-omic approach and include instrumentation for Droplet ddPCR, which initially was run on the QX200. And now we have added a next generation instrument, the QX1, that has expanded channels and automated workflows. We have NGS, currently being run on the Ion Gene Studio S5 Prime instrument. And then on the proteomics front, we have multiple types of MALDI-TOF from various manufacturers and multiple models, which allow us to discover, develop, and commercialize clinical tests. We also have LC-MS capabilities, as well as high-throughput automated ELISA systems. And then finally, we have a machine learning competency and proprietary platform that allows us to integrate large data sets and discover new tests. Here's a detailed list of our current offerings. We currently have a host of genomic and proteomic liquid biopsy capabilities for our various platforms. And this talk will focus on our next generation sequencing platform and the TCR Beta assay. So first, I'll give you some background on immune checkpoint inhibitors. So immune checkpoint inhibitors have provided unprecedented therapeutic benefit in the context of many different types of cancer. These therapeutic agents are employed in the clinic as mono or combination therapies and target negative regulators of immune activation using antibodies that block the CTLA-4 or PD-1 pathways. And by inhibiting the blockade at these receptors, T-cell activity is restored and immune cell response to tumor cells is enhanced. However, the response rate to immune checkpoint inhibitor therapy varies across patients and cancer types. Here I'm providing a few examples of the objective response rate to immunotherapy across different disease states. It's extremely effective, for example, in Hodgkin's disease, as well as in cancers with high microsatellite instability, such as Lynch syndrome. On the other hand, the response rate is as low as 20% in some cancers, including non-small cell lung cancer. In addition, a large percentage of patients treated with immune checkpoint inhibitors experience immune-related adverse events. This slide is showing some examples of adverse events reported across a selection of clinical trials for immune checkpoint agents. And as you can see, adverse events are actually quite common, occurring in as much as 45% of patients in the case of diarrhea in the Checkmate 067 study, which looked at the combination therapy of ipilimumab and nivolumab, with more severe grade 3 and higher events occurring in as many as 20% of patients, as was the case for hepatic adverse events in the same study. Current predictive biomarkers that are approved for use with checkpoint inhibitor therapy include tumor PD-L1 expression, tumor mutation burden, and microsatellite instability. None of these biomarkers on its own or in combination is 100% sensitive and specific for predicting responsiveness, and certainly not in all cancer types. So a host of other candidate biomarkers are currently being explored. Research into biomarkers for immune checkpoint inhibitor therapy is currently an area of great activity, and there are many promising emerging biomarkers currently being evaluated. Everything ranging from neoantigen load to clinical characteristics such as gender and age. So what I'm showing here is not meant to be a comprehensive list, but merely to highlight the amount of research that is occurring right now in this important field due to the critical need for effective biomarkers. So I'm sharing this diagram again because I think it does a nice job highlighting the critical role that T cells play in the response to immunotherapy. T cells are the effector cells, and thus the status of the repertoire as well as changes are indicative of the potential for response or response itself. The T cell receptor is a heterodimer of the alpha and beta chains, which binds to antigens presented by antigen-presenting cells, causing T cell activation and proliferation. It's estimated that there are on the order of 10 to the 14th unique T cell clones in a single individual due to somatic recombination of the genes encoding for the receptor and the addition of non-templated sequences during T cell maturation. The T cell receptor beta locus consists of variable diversity and joining genes, and the CDR3 region of the diversity locus confers much of the antigen specificity to the receptor. So the status of and changes to the T cell repertoire can be measured by next generation sequencing assays that target the CDR3 region of the TCR beta locus. A number of clinical trials have used T cell repertoire metrics as biomarkers for response to immune checkpoint inhibitor therapy. In the second to right column of this table of trials, you can see the metrics that are used as biomarkers in these trials. They include richness, Shannon diversity, entropy, evenness, clonality, and TCR convergence. And in this presentation, we will focus on clonality and TCR convergence. For our studies, clonality was determined from the evenness score and was defined as 1 minus evenness. Evenness expresses how evenly the clones in a sample are distributed among the different clonotypes. In this diagram here, each color represents a unique clonotype in a population of T cells. And then a perfectly even T cell population would contain equal representation of all clonotypes in the sample, and it's represented by the image on the left. However, a sample with low evenness and thus high clonality might, for example, look more like the population on the right, where the 3 different clonotypes are present at very different concentrations within the sample. TCR convergence is another T-cell repertoire metric, which is measured by looking at the percentage of clones in a sample that are identical at the amino acid level but differ at the DNA sequence level. It's thought to indicate T cells that independently underwent VDJ recombination but proliferated in response to a common antigen. So the frequency of convergent TCRs within a repertoire may provide an indication of the immunogenicity of a tumor and thus its sensitivity to immunotherapy. So I wanted to show just a couple of studies where T-cell metrics have been used or have been correlated with response to immunotherapy. So in this study, a cohort of 40 late-stage NSCLC patients were treated with anti-PD-1 or PD-L1 therapy, and their sorted PD-1 positive CD8 peripheral T cells underwent TCR sequencing. The population was stratified into patients with high versus low T-cell diversity. And as you can see in this Kaplan-Meier curve, overall survival was significantly prolonged in the high diversity group relative to the low diversity group. Importantly, the same pattern was not seen with unsorted T cells. And this study was looking at a cohort of 29 urothelial carcinoma patients. The investigators used T-cell receptor sequencing to assess the clonality scores in peripheral blood T cells prior to treatment with atezolizumab, and found that progression-free survival and overall survival were higher in patients with pre-treatment clonality scores that were higher than the median. And then finally, this study looked at TCR convergence and clonality scores in a cohort of 22 patients with mixed cancer types receiving CTLA-4 monotherapy. The investigators found that both convergence and clonality scores trended higher in responders relative to non-responders, and that a model combining the 2 metrics significantly discriminated patients with progressive disease from those with objective clinical response. So now I'll talk to you about our in-house TCR sequencing workflow. So in the Biodesix laboratory, we use whole blood and enrich for peripheral blood lymphocytes. And then from that buffy coat enrichment, we isolate RNA. And then once the RNA is isolated, it goes into our TCR beta sequencing workflow, which is run on our Ion Gene Studio S5. So this is a schematic of the Oncomine TCR Beta-SR assay, which targets the CDR3 region within the TCR beta locus, which, as I mentioned before, is the main antigen binding region, and thus a major contributor to T-cell antigen specificity. The assay itself consists of multiplex primers that bind to the framework region 3 and joining regions of the locus, producing amplicons approximately 80 base pairs in length for sequencing. One advantage of this design is that it allows degraded specimens such as FFPE or fragmented circulating nucleic acid to be sequenced due to its short amplicon length. This is an advantage that we've leveraged in our lab as we use StrectBCT to allow for shipping of samples to the laboratory at room temperature, which can lead to some nucleic acid degradation due to the fixative used to stabilize the blood sample. So here are the different outputs from the assay. So you get typical sample QC. You also get this really nice, just full list of clones, and each line represents a single clonotype with a specific variable and joining genes identified, as well as the actual CDR3 sequence, and the total number of counts and their frequency in the overall population, and then finally the rank for the clonotype. There's also these spectratyping plots, which is a visual representation of clone frequencies. And so each of these circles are bins containing all of the clones with a particular V gene CDR3 nucleotide length combination. So they'll contain multiple clonotypes potentially within each circle. And then the circle size indicates the frequency of all of the clones contained in the bin. And then it gives you various repertoire metrics, so values for evenness, Shannon diversity, the number of clones detected, and TCR convergence. So now I'll move on to the studies that we've performed with this assay. The first one being sequencing of peripheral blood leukocytes from NSCLC donors. So in this study, 5 different NSCLC specimens were tested using our TCR beta sequencing workflow. We didn't know the disease stage of the patients when we tested them. And we found that the quality of the RNA varied significantly across donors, which is what I'm showing here. We chose to use for this the DV200 score rather than RIN score because it was developed to measure the quality of highly degraded RNA from FFPE samples for RNA-seq, which makes it more suitable for looking at peripheral blood lymphocyte RNA from STREC-BCT because it tends, as I mentioned, to be fragmented following shipment at room temperature. And as you can see here from our samples, the DV200 score values ranged from 52 to 77%. So then, I would mention that one strength of this library prep workflow in particular is that it includes a functional RNA qualification assay, which is a qPCR-based assay that measures both the T-cell RNA content by measuring the T-cell specific CD3 transcript and the amplifiable quality of the sample because it's qPCR. So based on the results of the CD3 qualification assay, the RNA input ranged from 34 to 400 nanograms for the assay with these samples. So for donors 1 through 4, the really highly variable inputs resulted in consistent library metrics, about 1.5 to 2 million raw reads, around 70 to 80% usable reads, and pretty consistent numbers of clones from every sample. And however, for donor 5, the number of raw and usable reads, can't see the productive and rescued, but the number of raw and usable reads, which were 1.17 million and 63% respectively, was relatively low compared to the other samples. And the assay detected many fewer clones than the other samples, potentially due to lower input quality. So this figure, I'm showing the spectratyping plots for all 5 donors. And again, just to remind you, the circles are bins containing all clones with a particular V gene, which is on the y-axis, and CDR3 nucleotide on the x-axis length combination. And the circle size indicates the frequency of all the clones in the bin. And as you can see, the expansion of 2 different clones is visible in the spectratyping plot for donor 5. And this corresponds to a relatively high clonality score, as you might expect, which I'm showing here. Okay, so now moving on to our model antigen study that we did using cytomegalovirus antigen. So human cytomegalovirus is a very common immune evasive herpes virus that's found in all geographic locations and infects almost 100% of adults in developing countries and around 60 to 70% in developed countries. And we decided to use this as a model system for tumor antigenicity due to the fact that CMV is known to lead to clonal proliferation of T cells, much like tumor antigens do, and it's also a source of chronic antigen stimulation. So here is a diagram of what we did for the study. We took T cells in the form of PBMCs from 4 CMV seropositive donors, and we cultured them for 6 days in vitro in the presence of CMV whole antigen, which is called lysate from here on out, or the CMV immunodominant PP65 peptide. So, an entire mixture of peptides versus a single peptide. So we could see differences between the two. Then RNA was isolated at baseline from PBMCs prior to antigen challenge, and also after stimulation with either the whole cell CMV lysate or the single PP65 peptide. In addition to isolating RNA, we also prepped some cells for a PP65 tetramer flow cytometry assay, which we used as an orthogonal method to assess the percent of PP65 reactive cells in each of the conditions. So here are the RNA and sequencing quality metrics for the 4 different donor samples across the 3 different treatment conditions: baseline, stimulation with CMV whole lysate, and stimulation with PP65 immunodominant peptide. And as you can see in this table, a lower input of RNA ranging from 25 to 107 nanograms was required for these samples. So it was lower than the NSCLC specimens because of the high quality of the RNA that was retrieved from cultured PBMC rather than PBMC from preservative struct tubes. So the RIN scores here were 9.9 to 10, so very high quality RNA. However, this resulted in similar sequencing metrics to what was seen with relatively fragmented RNA from struct BCT. So this supports the idea that the assay is suitable for fragmented RNA samples, demonstrating its utility for testing of specimens collected in devices that stabilize blood for transport at ambient temperatures. In this figure, we are reporting on the TCR convergence scores across all of the conditions. And as you can see in the top panel, TCR convergence scores varied across donors at baseline and in response to antigen challenge. The highest TCR convergence score was observed in the peptide stimulated cells from donor 3. And when we drilled down into the clone table data, we found that this convergence score was driven by 2 sets of TCR convergent clones, which are shown in the panel at the bottom. You might find it surprising that convergence increased with such an acute antigen challenge as only 6 days. However, we believe that it's possible that the convergent clones were present at baseline at low frequency and were primed for clonal expansion upon antigen stimulation. Interestingly, I looked for the corresponding clonotypes in the baseline samples for donor 3, but didn't find them in the clone table. And this could possibly be explained by subsampling bias, and definitely highlights the importance of sample input and multiplexing as key variables to optimize during development of a clinical TCR beta assay. Here we're showing the clonality scores for the donors at baseline relative to the antigen stimulated samples. And as you can see in the graph at the top, clonality scores increased in the CMV lysate condition in particular relative to baseline for all 4 donors. And the increase was most dramatic for donor 4. And this clonal expansion can be observed in the spectratyping plots for donor 4 as well, which are shown in the bottom panel. And the most prevalent clone and driver of the high clonality score was observed at 76% frequency within the data set, and is represented by the prominent white circle indicated by the red box. This clone was also present in the corresponding baseline sample at 6.5% frequency, which is indicated by the yellow box on the left. So as I mentioned, we also looked at expansion of CMV PP65 reactive T cells post-antigen challenge by flow cytometry using the CMV PP65 tetramer reagent. This figure shows CD8 positive T cell populations from each of the donors prior to in vitro stimulation and after stimulation with CMV lysate or the PP65 peptide. In the T cell populations from donors 1 through 3, a very low abundance of tetramer positive CD8 positive T cells was observed prior to stimulation, while undetectable or marginal increases in tetramer positive CD8 T cells was observed after stimulation with lysate. However, stimulation with the peptide resulted in significant increases in tetramer positive T cells, as you can see in the column at the right. And donor 4 was a little bit different. This donor had about 13.7% of the CD8 positive T cells that were PP65 reactive prior to in vitro stimulation. And then with the lysate stimulation, the population expanded to about 26%. And then here I'm showing you the sequencing results for a single family of clones that we honed in on that just happened to be detected in all donors and expanded in response to stimulation with both PP65 peptide and the whole cell lysate. There are even multiple clonotypes for donor 2 that fall in this family, indicating there's a strong selection for this family of clones with the CMV antigen challenge. And then finally, we have shown that the expansion as observed by sequencing correlated very strongly with PP65 tetramer changes. So we compared what I'm showing here is a comparison of the fold change and the clone frequency for that family of clones in response to lysate challenge to the percent PP65 responsive CD8 positive cells. So the fold increase there by the flow cytometry assay for all 4 donors. And the R-squared for the linear regression analysis for this assessment was almost 0.94, which is a pretty impressive correlation considering the small sample size. We also observed something similar in the peptide stimulation condition for 3 of the 4 donors with an R-square of about 0.95. So pretty compelling correlation considering the sample size here. And that's it. In conclusion, the Oncomine TCR Beta assay detects clonal repertoire features as well as T-cell expansions with very high resolution using peripheral blood lymphocytes isolated from whole blood. We saw a really nice correlation of the RNA-seq method and the protein tetramer analysis using flow cytometry. And importantly, these techniques represent an emerging solution that could complement other liquid and tissue diagnostic assays in the clinic and will be of value as a predictive biomarker for immunotherapy. So we are currently pursuing studies to further evaluate the clinical utility of sequencing the T-cell repertoire in NSCLC patients receiving immunotherapy. So I'd like to acknowledge all of the team Biodesix, staying socially distanced in this picture here, as well as our collaborators at Celero, Benjamin Tioja, and Ann Lodge, as well as the team at Thermo Fisher Scientific for all of their contributions. So thank you so much.